Hello, everyone, and welcome to today's webinar, Learning to Aggress, from Complex Social Behavior to Whole Brain Light Sheet Microscopy, presented by Dr. Sam Golden and brought to you by Miltenyi Biotech. I'm Byron Hartman, and I'll be your moderator for today's event. Let's get started. Before we begin, I'd like to remind everyone that this is an interactive event. We encourage you to participate by submitting as many questions as you like at any time during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation during the live Q&A with Dr. Golden. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or you can report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I will now present today's speaker, Dr. Sam Golden, Assistant Professor at the University of Washington Department of Biological Structure. Sam received his PhD in neuroscience from the Icahn School of Medicine in 2015 under Dr. Scott Rousseau, and then he completed postdoctoral fellowship at the National Institute on Drug Abuse in 2018 under Dr. Yavin Shaham. Sam joined the University of Washington Department of Biological Structure in 2019 with a co-appointment as a participating faculty in the UW Center of Excellence in Neurobiology of Addiction, Pain, and Emotion. Dr. Golden's scientific interests encompass understanding the psychological and neural mechanisms guiding reward processing. He's particularly interested in understanding how neuropsychiatric disorders, such as maladaptive aggression, depression, and substance abuse, subvert basic reward circuitry to manifest pathological behavior. Currently, he aims to better define the intersection of aggression and motivation and identify the cellular and circuit mechanisms that control the transition from adaptive aggression to maladaptive aggression-seeking behavior. To investigate these mechanisms, the Golden Lab has pioneered new applications of approaches employing chemogenetics, optogenetics, calcium imaging, whole mount light sheet fluorescent microscopy, machine learning, and more. For a complete biography on our speaker, please visit the biography tab at the top of your screen. Also check out the links on the right, including a link to the Golden Lab website. So next, we're going to play Sam's pre-recorded presentation, and then we're going to follow that up with a live Q&A with Dr. Golden. Let's begin.
Well, first, I'd like to thank Miltenyi and Byron for inviting me to come give this talk. I'll say before we start that this is the first time I've given a lecture like this in this format, where it's pre-recorded at home and then followed by a live question and answer. And I'm very excited for the question and answer, but I am a little bit nervous to have to actually watch myself prior to that. So if we run into any technical difficulties, please bear with me, and I promise you we'll get to the end. So my name is Sam Golden. I'm an assistant professor at the Department of Biological Structure at the University of Washington. My lab focuses on the study of complex social behavior and its intersection with neuropsychiatric disorders, such as substance abuse and depression. But our key interest within that larger field is looking at this idea of aggression, reward, and motivation, and how that interacts with these neuropsychiatric disorders. So today what I'm going to talk about is how we model this idea of aggression reward in preclinical rodent models, and also one selective neural population that we've identified that seems to play a key role in regulating aggression reward. I'm also going to start talking about two other more preliminary projects, one on using machine learning approaches to analyze social behavior, and the other using whole brain functional connectomics to look at the pattern of brain activity that seems to underlie these states. And before I do that, I'd just like to thank the funding mechanisms that have allowed us to answer these questions, and even more so, acknowledge the individuals who are actually doing this work in the lab. So aggression is certainly not something that has been understudied. Going back to the 40s, 50s, and 60s, there's a really beautiful and elegant literature using electrical stimulation to identify what parts of the brain are responsible for the initiation of aggressive behavior. And if you took an intro to psych class, you might remember a video like this one, where we're seeing electrical stimulation of the lateral hypothalamus in a cat, leading to really robust defensive behavior. And so what these studies identified is that there's an area called the hypothalamic attack area, which starts in the most lateral portion of the ventromedial hypothalamus and extends back into the lateral hypothalamus proper. That really seems to be key for initiating this type of aggression. And this work really played a beautiful framework for more recent studies using optogenetics or chemogenetic approaches to identify the specific neural populations that are responsible. So this is really seminal work by Dayu Lin, where she showed that it was ESR1 alpha positive neurons within this most lateral aspect of VMH that, when activated, induced this really robust attack behavior. And so when this occurred, it really, when I first saw this video, it really captured my imagination. Because I looked at this and what we've come to sort of affectionately call the glove test. And what first popped into my mind was that the animal doesn't really have any emotional valence associated with this glove. And so optogenetic stimulation of this population is turning on or off the behavior. But in the real world, we often see that aggression is something that has a really high emotional or motivational state associated with it, whether it's a positive one or a negative one. And that question is really at the basis of the work that I'm going to show in the next 45 minutes or so. So aggression in mice itself is really interesting. It's highly stereotyped. There are rules that mice seem to understand when it comes to attacking another mouse. The face is off limits, the throat is off limits, the genitals and underbelly are off limits, and more often than not, the strikes are to the rear flank of the intruder mouse. But what's remarkable is that whether this aggression is due to a predator response or due to just attacking a conspecific for other reasons, it often looks really, really similar. However, even within a brain region like the VMH, we can see that the reason for attack can activate very different neural populations. So here we're using FOS, which is a marker of neural activity. I'll be talking about FOS a lot throughout this lecture, and so we use it because it allows us to identify neurons that were active during a specific behavioral window. And in this case, what we can see is that in response to a predator threat, the more dorsal aspect of the VMH is engaged, while in response to a attack on a conspecific, it appears to be the more lateral. And so this right away tells us that there must be dissociable mechanisms guiding different forms of aggressive behavior. And this has certainly been shown in humans. And this is an example from James Moran that I really like. It is a study done in healthy college volunteers where they were asked to listen to either appetitive or reactive auditory scripts of aggression or a control condition. And what they found is that there are really different patterns of cortical activity associated with these different types of auditory scripts that are asking these individuals to imagine themselves in an aggressive situation. This type of differential pattern is even more obvious or more identifiable when we move into the pathological forms of human aggression. So for example, repeat violent offenders have been shown to have both functional and structural abnormalities in limbic brain regions associated with their repeat violent offenses. And so the way that I try to conceptualize this study, looking at aggression reward or aggression motivation, is to do it within the framework of other really well-validated approaches. And I think one of the great examples of this has been the approach taken by the National Institute on Drug Abuse, which uses this idea of looking at drug use, followed by some form of abstinence and relapse. And I'd like to point out that even for all of the preclinical work that has been done using these models, there still hasn't been much of an improvement in relapse rates over the decades to numerous drugs of abuse. And I'm going to come back to why I think that's important in a second. So we wanted to take this idea and see if we could use similar preclinical models to look at this idea of pathological aggression. So being involved in aggressive encounters, going through some form of abstinence from that aggression, and then seeing if there is a relapse liability. And just as one example, with many caveats, but that I think really highlights why this is interesting, is if we look at recidivism rates for repeat violent offenders within the United States, we can see that almost 75% of violent offenders are back in jail within 5 years for repeat violent crimes. And of course, this is one extreme example that has many, many caveats to its interpretation. But I think it's much, much easier for us to just look at how enthusiastically followed sports like mixed martial arts and football are, to see that there are definitely this idea of reward and motivation involved with these aggressive encounters across all different spectrums of human behavior. And so the question is, can we reverse translate that? Can we go from this idea that we see all the time in humans and move it into a preclinical model so we can begin to identify the neural substrates and mechanisms that are guiding it? So luckily, there are some really great preclinical approaches to looking at stress-related behaviors. And I think one really classic one is resident intruder testing or chronic social defeat stress. So in chronic social defeat stress, we take a larger, more aggressive CD1. This is an outbred mouse. It is generally older and sexually experienced relative to the smaller subordinate inbred C57 intruder. This is a rigged game because of the size, age, and sexual experience difference between these two mice. The CD1 almost always chooses to engage in and win any aggressive encounters against the C57. And classically, we use this approach not to look at the CD1, but rather to look at stress susceptibility or resilience in the C57. And so this is what I did originally in graduate school. I spent many hours looking at videos of these mice beating up these mice, but to look at depression-related behaviors. But what I was anecdotally always much more interested in was the fact that we seemed to get a really even split of CD1s that were aggressive enough to be used as tools during chronic social defeat stress. And so in my mind, when Dayu's beautiful paper came out in 2011, and I was staring at these mice fighting for far too many hours, I really wanted to understand why that individual variability existed, why some were aggressive and some were not. And so we went ahead and screened a cohort of several hundred outbred CD1s for aggression over 3 days of resident intruder testing. And what we found is about 70% of any given cohort of CD1s exhibited some level of aggression across 3 days. But what was really interesting is that if you look at the mean latency over the 3 days between each of those aggressive mice, there's a really beautiful spectrum. And so now this allows us to look at the animals that have very low latency or very high levels of aggression, medium level or relatively weak aggression, and of course animals that fail to show any aggression at all. So using this type of individual variability, we can start to ask what's different. Why are some extremely aggressive? Why are some not aggressive at all? Unfortunately, with resident intruder testing, you can't really differentiate between reactive aggression and appetitive aggression. Or that is, aggression that's in response to a threat, that fight or flight response, or aggression that's because an animal finds it rewarding, because it's a forced encounter in their home cage. So to get around that, we needed to develop a new method that would allow us to isolate the initiation of aggression from the expression of reward. And to do that, we used aggression condition place preference. So in aggression CPP, you have a 3-chamber apparatus, a chamber on the left, a chamber in the middle, and a chamber on the right. And the middle chamber is neutral, but the left and right chambers have significantly different contextual cues. So for example, the size of the floor grids might be used, the coloration of the walls, the light gradient. Oftentimes labs will use an olfactory cue that's different between the two contexts. But the point here is that it's very easy for a mouse to identify context A and context B. And so using this type of apparatus, we could pair one side with a fight or an aggressive interaction and the other side with the absence of a fight or other appropriate control condition. And so we pre-screened aggressors, or CD1s, to find out if they were aggressive or non-aggressive, and then put them through this CPP procedure where they're contained to either context A or context B, and they can't get to the other side for several pairings. And then on the test day, we give them full access to the whole 3 chambers without an intruder present. And so then the CD1 can run around, and it can choose which side it wants to spend more time on. And if it chooses to spend more time on the fight paired side, we'd call that an aggression condition place preference. Whereas if it wants to spend more time on the unpaired side, we'd call that a condition place aversion. What we found was that the pre-screened aggressors form a CPP, while the non-aggressors form a CPA. So this was really exciting because this allows us now to begin to start looking at the neural mechanisms that are guiding not the initiation, but the motivation. We went on to do some other experiments to really better define this phenomenon of aggression CPP. And so I want to highlight three main findings. The first is that this is a very plastic behavior. So what we did is we took a large cohort of CD1s and put them through not 3 days of pre-screening to identify if they're aggressive or non-aggressive, but 10 days. And what we found is that over the course of these 10 screening sessions, starting with animals that were non-aggressive or aggressive, roughly 50% of the non-aggressive mice stochastically switched to an aggressive phenotype. More importantly, once they switched, here these animals that switched are indicated in gray, they went on to form a conditioned place preference to the fight-paired side. So these were mice that were previously non-aggressive, became aggressive, and then went on to actually form a conditioned place preference for the fight-paired chamber. So there's behavioral plasticity here. It can be learned. And it's also quite persistent. If we test the same animals for CPP several weeks later, we find that the aggressors still prefer the aggression-paired context, and the non-aggressors still prefer the intruder-unpaired context. And again, those that switched look like your standard aggressors. So there are some limitations to using CPP. A major limitation is that we still can't appropriately differentiate between reactive and appetitive aggression because, again, it's still a forced encounter. We're still forcing the CD1 into context A or forcing them into context B. And so luckily, there has been a solution already identified in the literature, which is operant aggression, or training a mouse to lever press for access to a subordinate intruder. And this is some really beautiful work that was initially done by the Mitchell lab in the early 2000s, where they showed that outbred mouse will actually nose poke for access to a subordinate intruder. And so what we wanted to do was take this operant approach and make it higher throughput, and then apply some of the gold standards from the addiction literature, but within the context of aggression reward seeking. So there are certainly some core features that are commonly held to indicate an addiction like rodent. These are, for drugs, these are persistent relapse during abstinence, a very high level of motivation to seek out, or amount of effort they'll expend to seek out the drugs, and continuing to work for the drugs even in the face of severe adverse consequences. And so the question was, if we took our aggressive animals, our aggressive CD1s, and applied these same metrics using operant procedures, would we be able to identify any mice, any aggressive mice, that meet or exceed the commonly held criteria for addiction-like behavior? And so this is how we actually do this. We have operant chambers, just like operant chambers you'd use for drugs of abuse. In our case, though, we have two active levers, one on the left side here, one on the right side here. The active lever on the left is paired with something, with whatever we like. In this case, it's a highly palatable food reinforcer, but it could be alcohol, it could be other drugs of abuse. The lever on the right is paired with an automatic guillotine door that allows access to a subordinate intruder. To make this high throughput and amenable to the types of neuroscience techniques that we're commonly using, we have a multi-mouse magazine for the C57 intruder. So there's actually a mouse on the bottom and a mouse on the top, and when the automatic door opens, we can push in the intruder, and then we can cycle them rapidly so that one individual can run a bunch of boxes at one time. And here's what it actually looks like. So in this case, what we're looking at is a variation on self-administration where there are two active levers in the box. The first active lever needs to be pushed 10 times. Once it's pushed 10 times, the second active lever becomes available, which only needs to be pressed once, which leads to the opening of the automatic guillotine door and access to the subordinate intruder. The CD1 does all of this voluntarily. In this case, they have to work quite hard to do it. They need to go through a chain of events to gain access. And then they also get to choose what they want to do when the intruder is present. So in this case, after about 10 seconds, the CD1 chose to attack. So using this approach, we can now look at a completely voluntary form of aggression motivation. When we do this over several days, we see that they learn to self-administer quite well. We keep track of two metrics. Was it a reinforced or rewarded trial? So did they press the lever that gained access? And also, once they had access to the intruder, did they choose to engage in an aggressive interaction? We separate the mice as soon as the first aggressive bout is done to make sure that none of them are harmed. And this also allows us to get between 12 and 20 trials during each day's training. And so in this case, what we can see is that they acquire the lever pressing for aggression, and that they attack on the majority of trials when they do lever press. So this was very exciting because it gave us a high throughput, very operationalized procedure to look at operant aggression self-administration. But then again, we wanted to come back to this idea of relapse liability. And so we started with forced abstinence, which is very simple. It's where you move the animal out of the environment, out of the operant chambers, and then re-expose them under extinction conditions at some later time point. And here we chose either 1 day or 15 days after the end of training. And what we saw was a strong, time-independent relapse to aggression-seeking behavior. Of course, this is not one of the gold standards that I just presented, but it was very exciting for us because it suggested that we should be moving in this direction. And so we then went ahead and actually did apply all of these other core or cardinal approaches for identifying addiction-like behavior in rodents. So we took, for example, a very large cohort. We put them through self-administration, early and late relapse, something we call voluntary abstinence. So in voluntary abstinence, we take advantage of the fact that we have 2 active levers within each operant chamber, and we train the aggressive mice to self-administer highly palatable food under non-food restricted conditions, and also to self-administer aggression. And then we give them a choice. We give them a series of choice trials where we can say, which would you prefer? Would you prefer access to highly palatable food, or would you prefer access to a subordinate intruder? And what we find under these choice parameters is that the vast majority of aggressive mice will choose food over fighting. This type of voluntary abstinence is how we can model something like contingency management from the clinic. So under contingency management, a human who is coming in will be provided a voucher, maybe for money, maybe for a job, in exchange for providing clean urine. And this type of approach is highly effective at attenuating drug seeking in humans. And this is how we can actually try to use a similar approach in preclinical rodent models. We also put them through progressive ratio. So progressive ratio is a procedure where each subsequent reinforcer requires a higher level of effort than the previous. And it's not an exact exponential, but it's essentially like 2, 4, 8, 16, 32. And at some point, the mouse will say, this is just far too much effort to expend to get this reinforcer. And so it gives us what we call a break point, or the point at which they will no longer continue working for that reward. And we also used punishment. So under punishment conditions, 50% of all reinforced lever presses are paired with a foot shock. And so you can imagine that as this contingent foot shock occurs, the mice learn very rapidly to no longer lever press for the reward, in this case aggression. And so we put this large cohort of CD1s through all of these procedures so that we could look for any subpopulations that might develop. What we found using cluster analysis is that this is unsupervised cluster analysis, is that we find 2 different populations of aggressive animals. One we term typical aggression seeking, and another we term compulsive aggressive seeking. So the compulsive aggressive seeking population is roughly 20% of any given cohort. And if we reanalyze all those individual data points now by these new groups, what we find is that the compulsive aggressive seeking animals are just off the charts by any of the metrics that we use, whether it's attacks, relapse, choice. So again, most mice will choose food over fighting. These guys will choose fighting over food. Breakpoint. To put this in perspective, a breakpoint of 20 reinforcers is roughly 600 lever presses just for that most recent reinforcer. So they're working incredibly hard for access to fight. And they're completely punishment insensitive. So this is really interesting because now we have an opportunity where we can look at totally voluntary aggression self-administration along with all of these forms of relapse, forced relapse, voluntary abstinence, or punishment-induced abstinence. But also we can identify subpopulations that meet the criteria for compulsive aggressive behavior. So we can look at the typical mice, the compulsive aggressive mice, all under these completely voluntary conditions. And so using these types of approaches, this slide really summarizes what we know about the neural mechanisms of appetitive aggression. One story that I'd really like to highlight now has recently been published is looking at the role of the nucleus accumbens. So the nucleus accumbens is a critical brain region within the reward circuit. It's maybe thought of as the interface between motivation and action. It's composed predominantly of what are called medium spiny neurons, and they come in two flavors: D1 receptor expression, or dopamine 1 receptor expressing, or D2 expressing neurons. They make up about 95%. There's also interneurons. If you think back again to maybe that intro to psych course, the D1 population is often called the direct pathway and the D2 indirect pathway. So what's really interesting about this is it's been critical or identified as a critical brain region in other types of natural and non-natural reward as well, such as drugs of abuse. And so we wanted to identify if it was also involved in aggression, self-administration, and relapse. And so again, we used FOSS, that marker of neural activity, to look in the nucleus accumbens and identify if it was active during either reinforced aggression or relapse compared to control groups. And what we found is that under both reinforced conditions and aggression-seeking conditions, we have a strong recruitment of the nucleus accumbens shell. Like I said, it's made up predominantly of two different cell types, the D1 and D2 expressing. So our next question was, one, could we recapitulate that general activity, which we were able to do, but also, is there a specific population within the accumbens, the D1 or the D2, that are really engaged by this behavior? And what we found was that both the D1 and the D2 MSNs were equally FOSS positive during either reinforced aggression or relapse to aggression seeking. So this wasn't what we were anticipating, but it was very, very exciting. Unfortunately, to causally identify the role that D1 and D2 neurons are playing, we need to have access to genetic tools. And as I mentioned, we tend to use these larger, more aggressive outbred mice, these CD1s. And outbred mice are not available with these Cree tools. So what we had to do was come up with a strategy that would allow us to isolate the genetic populations of interest. We did this through a transgenic hybrid breeding strategy, where we can take our D1 or D2 Cree inbred mouse, cross it with our outbred CD1, and take the outbred F1 hybrid population that now contains the D1 or D2 Cree transgene that we need to use to isolate these populations. This actually, gets us a completely novel new cross, and so we need to go through and developmentally and biochemically validate these crosses and make sure that they still show the behavior that we want and don't have any off-target molecular effects as well. What's very interesting is these transgenic hybrids are about the same size over the course of development as the CD1 and much larger than the C57 parent. And I'm not showing it here, but they also show very high levels of aggression, whether it's reactive aggression in resident intruder or operant aggression in self-administration. And so what we could then do now with these F1 hybrids that have Cree in either the D1 or D2 population within the nucleus accumbens is use chemogenetic approaches to artificially inactivate these populations during either self-administration or relapse behaviors. Again, we targeted the nucleus accumbens shell because that's where we identified the largest population of FOSS reactivity. When we do this, what we found was even though both the D1 and D2 populations were FOSS positive, only the D1 population seems to be playing a critical role in controlling both reinforced aggression as well as relapse to aggression-seeking behavior. So now we can say with some confidence that there are cell type specific differences in appetitive aggression seeking. So I just want to now give you a better example of what this aggression can look like. So I mentioned that we keep track of two metrics. Was it a rewarded trial? So did they press the lever? And was it an attack? So did they attack, binary yes or no? But if you look at this video, you can see that there's really a lot more going on. There are tail rattles, lateral threat displays, all different types of attacks. The intruder can be exhibiting different types of active or passive coping behaviors in response to these aggressions. And we don't get any of that in these experiments. And the reason we don't is if I ask someone in the audience to volunteer to manually score frame by frame hundreds of hours of these videos, I don't think I'm going to get a lot of volunteers. It's incredibly time demanding. It's prone to observer drift and observer bias. And it's one of the main reasons that complex social behavior is not often incorporated into many of the preclinical models that we use to look at neuropsychiatric disease. And so we wanted to see if we could maybe overcome that with some of the tools that have recently become available. So in this example, all of the behavioral annotations that you're seeing read out on the left side are automated. And that is, this video has never been scored by a human. And we can do this using machine learning-based approaches. Obviously, this is something that I think many graduate students and undergraduate students, you can hear their sigh of relief seeing a video like this, because this is really, again, a tremendously subjective and time-consuming process to do manually. So we wanted to take advantage of recent advances in pose estimation, which are basically, again, machine learning approaches to identify specific features in a video and apply them with predictive classifiers of these complex social behaviors. And to do this, we came up with a software that we've called Simba. I'll talk about it in a little detail in the upcoming slides. And this is the team that has really poured their heart into developing this open source pipeline. But what Simba allows us to do is make these types of predictive, complex social behavior predictive classifiers without any specialized hardware. We also wanted to be very clear that we could have transparency and explainability of the models we're working with. Again, many behavioral neuroscientists do not come, myself included, with classical training in computer science. And many of the machine learning tools that exist sort of operate within a black box. And that makes it really challenging to troubleshoot them or trust them. And so what we wanted to be able to do was bring transparency and explainability to this process, as well as have checkpoints for human validation so we could make sure everything was going well and correct it when it's not, and again, not require programming or computer science experience. And so it's very simple. It's just this very simple process. Okay, it's really not that simple. What Simba basically allows us to do is walk a behavioral neuroscientist or any other user through the process of processing videos, generating or importing a pose estimation behavioral tracking model, building classifiers of these behaviors. For us, it might be attack. For someone else, it might be grooming. And then lastly, actually analyze those and create visualizations. I just want to point out Simba is 100% open source. Simba stands for Simple Behavioral Analysis. So all of the files that were used, all the code that was generated, as well as all of the videos or files that were used to create either the tracking or predictive classifiers are openly available. And you can feel free to screenshot this and go check it out. And so coming back, I'm just going to sort of slowly walk us through each of these, just at a superficial level. This is all published as a preprint, so if you're interested in any one component, you can go ahead and pull the preprint and get the details. So the first step is obviously pre-processing video. It seems like a pretty straightforward thing, but when you have hundreds of videos acquired over the course of an experiment, it can actually be quite daunting. So what we've done is we've gone through and provided a pipeline that allows for either processing a single video or a whole batch of videos with cropping, trimming, changing colors, enhancing, various filters, things of that nature. But basically getting everything processed to the point where it's now possible to move forward with analysis. The second component is actually importing the tracking model. So these pose estimation architectures that I'm talking about are things like Deep Lab Cut, which is very popular, Deep Pose Kit, Sleep. Each of these use neural networks to identify various features on the mouse, such as the nose, the ears, the tails, the flanks, whatever you like. And then we can take those identified features, their XY coordinates, and use them to create the predictive classifiers. So one of the things that we thought would be really critical to get adoption of this type of machine learning approach into the behavioral neuroscience labs is making it really flexible. So as you can see, we tend to use a lot of annotations. We like to have the nose, the ears, the flanks, the tail base on both mice. But for other people, they might only need 4 of these body ports. And so what Simba allows us to do is flexibly utilize any of these any of these pose estimation annotation schemes, and then use them in the next steps. More recently, Deep Lab Cut has released a multi-animal module, and Leap has released their social Leap module that allows us now to work with similar looking mice. And so we've actually updated in the most recent release a few weeks ago of Simba the ability to use these types of pose estimation schemes as well. Right now we fully support 2 mice, but we're slowly rolling in this N+1 where we have up to 5 mice. So once you have your pose estimation, that's when you really are, now you've gone through the basic process of prepping your videos, getting your tracking, and now you're really into the heart of Simba, which is generating predictive classifiers. And so we have project management that allows us to calibrate your videos for distance and time, that allows us to correct outliers, extract features, and of course, go through all of the validation and optimization and explainability steps that I insinuated earlier. So what does that actually look like? So again, this is all GUI driven so that it doesn't require any programming experience. But first we remove any pose estimation that was inaccurate or is impossible between the multiple animals. We are also then annotating any regions or objects within an environment that will be really critical or crucial for incorporating as features within our machine learning pipeline. I also just point out that even though it wasn't our intention, it's become very popular to use Simba, not for machine learning purposes, but just for getting really thorough descriptive statistics of pose estimation. And certainly that is possible using the Simba pipeline as well. So once you have your pose estimation, you've corrected any outliers, you've added any regions of interest, you actually need to go through and identify the behaviors that you want to be able to automate the classification of. So that's what you're seeing here in this video is just an example of walking through the GUI. You can select a video, the video pops up. You can then annotate the specific behaviors that are present on any given frame and use those to develop the relationships between the pose estimation features that encapsulate the behavior that you want to find. And so this is what it actually looks like on the back end. As the animals are interacting, since you've behaviorally annotated the frames of interest, whether it's aggression or grooming or lateral threat display, we are then identifying the relationship between those pose estimations and all these possible combinations of other relationships. The reason we want to do this is then to be able to say in a new video with high accuracy and high precision that these behaviors are present or not. And we can do that at levels that are above your typical undergraduate, but certainly at or above a well-trained scorer. So when I come back to this last bit on the ability to evaluate these classifiers, this is, I think, really a critical component because without the ability to make sure they're working as intended and easily correct them when they're not, it becomes a very harrowing and challenging job to create really well-validated classifiers. And so we built this into Simba so that you can automatically generate not just the ones I'm going to show you, but many, many different types of evaluations. Precision recall is one that's very popular because it tells us what is the optimal discrimination threshold. So there's always a range of probabilities that an event has occurred, and you need to set a threshold when it is a binary yes or no. What's the optimal threshold to use? Do I need to add more training data to the training data set to be able to get these classifiers? You can identify that with learning curves. We also use Gini importances and permutation importance to understand how important or predictive any given feature is within these classifiers. And so all of this is generated and made available so that we can go in and understand what's occurring. And of course, you can get the actual prediction trees that were utilized as well. So the final step is going in and actually analyzing the data. There's many, many tools that we have available here, but I think the ones that people like to see are the visualizations. And so you can go in generate overlays, pick the parts of the data frame that you want to go along with that, and incorporate it into your analysis. So now what we can do is we can come back to examples like this, where we previously only had rewards and lever presses, and now go back and ask, during D1 inhibition, how are these behaviors actually shifting? And get a much deeper dive. We're also in the process of aligning these types of automated approaches with fiber photometry and calcium imaging. And I think more exciting to myself is moving forward with unsupervised analysis of the previously identified supervised epochs. So as an example here, what we call attack, or what we believe is an attack, could actually, here indicated in red, be made up of several different motifs. And so now we can go in using unsupervised analyses from SIMBA and begin to tease apart the individual components of these more complex behaviors as well. And so all of that is ongoing work. So the idea there is to bring behavioral neuroscience into this big data field, which is my hope we're going to be able to pair with some of the functional connectomics that I'm about to speak to you on. So whole brain functional connectomics gives us the opportunity to now ask not just what brain regions we have already identified and what the role is in aggression-related behaviors, but also look at all the non-canonical regions that have yet to be identified. We do this through brain clearing and then imaging with light sheet fluorescent microscopy. This is the work of postdoc in my lab, Eric, in collaboration with Tarun at NIDA. They've really been the heart behind this project. But what we can do is basically take a brain, make it transparent, and then image it as an intact volume without having to cut it up. So what that allows us to do, as an example, are get these really high-level, really nice renders, 3D accurate renders of whole intact brains. Here I'm obviously just showing in this example a thin 300 micron slice of a whole intact brain. In red is FOS, again that marker of neural activity. In this case in green is Thi1-GFP, which is just a marker of glutamatergic neurons. And we can go through, generate these volumetric data sets, and then identify the brain regions that are most active in these intact brains during our behaviors. This is what it actually looks like when we're looking at a brain analytically. Each one of these is a FOS-positive neuron. I'm not going to get into it now, how we go about the registration and segmentation of these data sets. But once they are registered and segmented, we can go in and start really figuring out the ensembles or patterns of brain regions that are active. Here I just want to show you what it actually looks like. So on the top is a no-test control. On the bottom is a relapse to aggression-seeking brain. And even by eye, you can see that there's certainly enriched patterns of FOS in the day 15 relapse. So what we're in the process of doing now is going back, finding all of these FOS-positive neurons, and then performing the analysis that allows us to understand which of these ensembles are engaged by reinforced aggression, by relapse to aggression seeking, and also by other behaviors. So before closing here, there is one new preliminary data set I'd really like to share, which is not on aggression, but which I think is very exciting, which is looking at the whole brain functional connectome of incubation of craving. So incubation of craving is a phenomenon that was first identified by Jeff Grimm and Yavin Shaham in the early 2000s, which is that the longer an animal is abstained from a drug of abuse, when they're reintroduced to the context where that drug is available, they will work harder for it. So this is called incubation of craving, because the longer the animal is abstained, the more they crave that drug. This was first shown with cocaine, but it's also more recently been extended to other classes of drugs, and has also been shown in rats for sucrose. What we've been working on recently is looking at highly palatable food self-administration and incubation at day 1, day 15, or day 60. What we've observed is that behaviorally, we do recapitulate this incubation of craving phenomenon for highly palatable food at later time points. And so now the question is, what areas of the brain are engaged by this incubation phenomenon? So here I'm just going to show a video which summarizes some of the differences and patterns of FOS immunoreactivity that we observe. On the top left are home cage controls, and then on the top right are day 1 relapse, bottom left day 15, and bottom right day 60. And what you can see just by eye is that at the later relapse points, there's a strong enrichment of FOS relative to earlier abstinence. So here I'm showing that data summarized. On the left is just going in the pattern of most FOS to least FOS across all experimental groups, and on the right, the home cage control. We can see that there are distinct differences or patterns of FOS enrichment. If we reorganize those by experimental groups, what we begin to see is that during incubation, there's a strong enrichment, not necessarily of unique brain regions, or not really onlining of unique brain regions, but rather a sensitization of brain regions that have slowly been ramping up during that time, during that abstinence period. And of course, we do see some significant regions even after correction for multiple comparison, many of them within the cortex and the hippocampus. And so right now, this is very preliminary. We've only completed collecting this data several weeks ago, and we're still crunching through it. But we think this is really exciting because it's an example of an application for whole brain imaging where we now have a very clear answer to a question that's existed since the behavior was first discovered. Does incubation involve the recruitment of new brain regions at later time points, or the sensitization of similar brain regions across time? And I think now that at least our preliminary data strongly suggests it's a sensitization of brain regions across time rather than a recruitment of novel ones. And so to give you an idea, here is this ensemble of brain regions that we've identified. And so now the goal, of course, is to take this approach, like we've just shown you here for incubation, but apply it to reinforced aggression and relapse to aggression-seeking behavior in combination with this machine learning approach to behavior. So we have big behavior, big imaging, and we'd like to pair them to really try to understand how these various brain regions are relating to the more complex components of these behaviors. So with that, I'm just going to say that Simba is available currently, and anyone who would like to go and give it a try, please do. And I'd also like to point out that although I didn't speak about it, we also have ongoing projects looking at the intersection of PTSD and maladaptive aggression and opioid withdrawal and escalated aggression. With that, I'd just like to once again thank the members of my lab that have really, of our lab, that have really made all of this work possible. And I think we will now transition to the live portion of the question and answer.
Thank you, Professor Golden, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, so the first question. In bipolar disorder, the patient will have an altered mood swing. What will be the brain activity and aggressiveness maximum limit in these patients? Can you comment on this question, Dr. Golden?
I can try. So obviously all the work that we do is in preclinical models. Is the sound coming through okay? I just want to make sure.
I think so. Yes, I can hear you quite well.
Okay. If you can hear me, then I think we're good. Yeah, so we can't really comment too much on what's happening in a clinical sense here. What I can say is that there is a clear need to understand the intersection between maladaptive social behavior like aggression, and which is clinically often represented more as irritability or agitation in these contexts, and how those do connect with this basic neurocircuitry that guides aggression itself and the intersection with motivation. We have not really explored that yet in our preclinical models. We certainly don't have a model of bipolar disorder. Some that we are working on, which I think are related, are, and I mentioned in our closing slides there, are projects looking at the intersection of PTSD and either escalated or attenuated maladaptive aggressive behavior. And it's too soon to really get into any preliminary data that we've identified on those in terms of the neuroanatomy. So I can't really speak to that yet, but it's something that's definitely on our radar. And those are the types of questions, of the translatable questions that we want to begin to address with these approaches. But it's early days, and we're still very much in process on this.
Thank you, Dr. Golden. Next question. Is there any predictive value at the beginning of your training with the 3-chamber test? So if you would do the 3-chamber test at the start of the training of the CD1, could you predict which ones will become aggressive, even if they're not aggressive at the initial start of the test?
That is a really good question. And it's one that we are actively looking into now. So Nastasia Goodwin, her ongoing project right now, one of them, her main one, is a very ambitious attempt at tackling that. What are the similarities and differences between reactive and appetitive aggression neural circuitry? And so what we can tell you anecdotally so far is we always pre-screen our CD1s at the beginning in either some type of resident intruder task or simple magazine training where we introduce them to the operant environment and do some Pavlovian conditioning to the cue and the presentation of an intruder. And the aggressive mice tend to go on to lever press very nicely for appetitive aggression. What's interesting is, at least anecdotally, is it's not terribly predictive of the ones that show a non-aggressive phenotype under those reactive conditions. So what I mean is, even animals that might pre-screen as relatively non-aggressive in a resident intruder can still go on to become very good lever pressers for appetitive aggression. And I think that ties in somewhat to the data we showed with our aggression CPP of there being a really nice behavioral flexibility or plasticity once animals start to win and continue to have access to winning interactions. So what could I say? So to answer your question a little more specifically, if an animal is aggressive reactively, they tend to acquire appetitive aggression well. If they are non-aggressive under reactive conditions, it is not anecdotally highly predictive of how they'll perform under appetitive tasks. But I think that's a super interesting observation, and it's one we're really following up on now. And hopefully we'll have some more answers in a year or two.
Excellent, thank you. The next question. Hi, Dr. Golden. Thank you so much for your presentation. This is a bit of a specific methods question. In your aggression CPP paper, it says 10 minutes in each context, intruder paired versus unpaired. In the intruder paired context, did you ever consider 5 minutes alone first so that the mice develop stronger territorially to that side before the introduction of the intruder?
Yeah, it's a good technical question on how to design and execute a CPP or other type of social context task. So we did not do that. That was not one of the permutations on the procedure that we played around with them. Housing them overnight the night before, for example, on the aggression paired side or anything of that nature. I have no doubt that could absolutely influence how the animals will behave. And so I think it's worth considering or certainly being aware of in designing these types of procedures. But we never specifically addressed that. Territoriality, especially in mice, is obviously a key driver of reactive aggression. We haven't, at least in our lab, looked much more in more detail into how territoriality interacts with aggression reward. But it's a very interesting question to think about.
Thank you, Sam. Next, thanks for the great talk, Dr. Golden. I have 2 questions. 1, from a social defeat standpoint, how do you think the individual variation in CD1 aggression affects the ultimate vulnerability or resiliency of the intruder mice? Is the variability in CD1 aggression one of the reasons for the split, aka can you shift intruder mice to resilient or susceptible based on latency and the amount of aggression? That's the first question. I'll let you answer that one first before we pose the second.
Yeah, so I mentioned that a lot of our conceptualization of how we've been looking at aggression really derives from the much larger and much more fleshed out chronic social defeat stress literature.
Right.
And so this is sort of a segue, not really relevant to the data I showed, but it's a really good question. Over 10 days of chronic social defeat stress, when you're using 10 different aggressors that are getting rotated each day with each of the intruders, so you're not having the same pairings on each subsequent day, there's going to be variability in the levels of aggression that any given intruder is exposed to. And there's no doubt that that type of variability can influence ultimately a susceptibility and resilience. There's no way around that. It takes two to tango, and we can't control the level of aggression that a CD1 shows during a social defeat procedure. We can certainly remove CD1s that are not showing aggression, and that's done very regularly in these procedures. But it gets to a much more interesting point, which is what are the intruders doing in response to the aggression that's being delivered? Are they using active or passive behavioral coping strategies? So in the C57, they could be going like this, is my interpretation of them showing a subordination. Or they could be turning tail and fleeing under more active conditions. And I think that is really the future of understanding the molecular mechanisms and circuit-based mechanisms that we're finding. We can't control how much aggression they get. We can certainly begin to categorize in much greater detail using SIMBA, using JABA, using unsupervised approaches. There are so many that are coming out now that need to be evaluated and incorporated into these types of behavioral procedures. We can use those now to separate in animals that use active versus passive, in animals that got high levels of aggression versus moderate levels of aggression over the 10 days. The differences that we're seeing. And I have no doubt that we will begin to better be able to predict under those types of analyses animals that ultimately go on to show susceptible phenotypes or resilient phenotypes. Because this is a behavior, and there's a cause and consequence at play, which we just haven't had. I mean, I certainly have not had the ability to really break down in a very granular way. I think the tools are now available and easy enough to incorporate in really normal, not computer sciencey behavioral neuroscience labs to start addressing those exact questions. And so I think I answered that. I think I'm very long-winded answer. Yes, I think the behavior of the CD1 plays a very important role in the behavior of the C57.
Excellent. And the follow-up question from the same viewer was, from your SIMBA behavioral analysis system, can we add our own behaviors on top of the tail rattle, for example, to predict our own features during an aggression session?
Yeah, so I guess I didn't make that clear enough, and so this is a very good question. SIMBA is agnostic to the behavior that you want to identify. It's supervised machine learning. You're supervising it to identify the behaviors that you want. We've released in what's already available a set of what we think are core behaviors for resident intruder and chronic social defeat stress on both the part of the resident and the part of the intruder. So allo grooming, under different vigorous versus less vigorous conditions, attack. If your tracking is very good, if your pose estimation is very good, things like tail rattle. We also include lateral threat display, fleeing, pursuit, all of these. But that's just the ones that we have a personal interest in the lab, and obviously those are going to be very different across labs. And so what SIMBA allows is the ability to go in and generate your own classifiers for your own behaviors of interest. And if you already have annotated videos, like with JWatcher or Boris, or if you like Noldis with EthoVision Observer, you can take those already annotated data sets and feed them into SIMBA and not have to go through and behaviorally annotate your videos. We've also incorporated a bunch of tips and tricks, such as pseudo-labeling, that allow you to much more rapidly identify the behaviors of interest that you're looking for than if you're just sitting there with a stopwatch and an Excel sheet. And so we're really trying to make that painless. Now, the reason, though, that we really think this is important isn't just as a tool to, in your own lab, or in your own experiments, identify specific behaviors. What we really want to be able to do is create a very common language of what I call attack and what you call attack, so that when we look at the molecular or circuit mechanisms that we're trying to associate or trying to say play a causal role in these behaviors, we're actually talking about the same behavior. That's obviously something that is not occurring right now in behavioral neuroscience. And oftentimes even the ethological breakdown of what someone is calling behavior is even presented in the methods. And so our hope is that by using these types of automated approaches where I can give you my attack classifier, you can give me your tail rattle classifier or whatever, not only will we then be using the exact same behavioral language for the comparisons we're making at a higher level, but as we build on them or as they change, there are metrics we can use to very clearly identify, not just observationally, but mathematically, why they're different or similar. And so I didn't get into that because I think that's a much more advanced use of Simba, but it's certainly something we're really excited and really digging into on our end and trying to incorporate into the GUI so that it's easily accessible to everyone as we try to make new classifiers, disseminate them, and share them among labs.
Excellent. Thanks a lot, Sam. So I'm going through the questions, and we still have quite a few. I think we'll stick around for a little bit while longer.
Oh my God, there's
I think
a lot of questions.
We've got a lot of questions. So, but I wanted to, I skimmed through and I looked and I noticed that there's been quite a few questions relating to how you take the whole brain imaging data and, interpret and register and segment that. I think maybe it'd be helpful if you were to briefly comment a little bit on the whole brain data, how you take that from raw data to analysis, and what that has done for your research program.
Everyone buckle up. It's going to be about another hour. No,
okay, let's try to keep it short.
Yeah, so there are so many amazing tools in development or that have already been released that can aid people going from these whole brain data sets to a registration and ultimately segmentation. What we're using currently is a combination. We use a multi-model approach where we use ClearMap, developed by Christopher Kirst, who just opened his lab at University of San Francisco like a month and a half ago. I'm sure he'll be looking for postdocs and students shortly. So if this type of thing interests you, I would look him up. We use ClearMap as our first base. It's a fully automated voxel-based system, which basically registers our samples to the Allen Brain Atlas Common Coordinate Framework. But that is a black box. And so a lot of times what we find during our clearing and imaging process is some of the brains get damaged or they're not quite as uniformly shaped as we would like for these automated processes. So we've incorporated a second system, which we call Whole Brain Smart, which there's a preprint for as well. So basically there's a beautiful R-based package called Whole Brain, which written by Daniel Firth, which allows for spline-based manual, semi-manual registration of whole brain images, again, to the same Allen framework. But what you can go in is actually add individual annotation or correspondence points on the interior of your images so that you can find your histological landmarks, even in a damaged brain, and get it mapped on well enough. And so one of our students back at NIDA wrote a great program called SMART, which basically makes it really straightforward using console-based commands rather than command lines to take a large data set, like a 4,000, 6,000, 8,000 image data set for a whole brain and put it through the whole brain pipeline. And combined using these first fully automated approaches and then semi-manual approaches to make corrections when things don't work has been working really well. And so that's sort of our general approach. Now, that said, I cannot speak highly enough of the many programs that are currently trying to push us even further towards full automation. CellFinder, developed by Adam Tyson, comes up in mind most recently as an approach that looks incredibly promising for these types of analyses. There's, what else? ClearMap 2 just got released. We're not as familiar with that, so I can't speak to that yet. Miracle, developed, I believe, in collaboration with the Deiseroth lab out of Stanford, is another approach which looks very promising. And so I mean, there's no one, of course, there's the cubic system as well, which has some beautiful things coming out, I think, in the next month online that are also going to allow these types of approaches. So there's a lot. There's no one simple answer for how do you register and how do you segment. Do you do it using machine learning approaches? Do you use it just using setting basic thresholds for different criteria for what a FOS positive or cell is? And I guess the take home here is people need to find the one that works the best with their pipeline. I would like to think there will be a more unified approach moving forward. We're not there yet, in my opinion. It's sort of a wild west, to be honest, right now. And again, the keys here, in my mind, are the same ones that we tried to employ in Simba, which is explainability, transparency, and the ability for humans to come in and validate at key checkpoints. Because a lot of stuff still is happening as a black box. It's great for usability. It's less great for having a lot of faith in how robust the data is that we're getting out. And so I think whatever pipeline ultimately does become the most standardized pipeline will have a lot of those checkpoints that allows the user to go in and actually see how good the registration is, make modifications to the registration as needed, see how good the segmentation is under different parameters across the whole brain. And then whoever creates this dream system will be very popular. Let's put it that way. If you really, really want the details of how we do this, I suggest you check out the SMART preprint that's up on bioRxiv. I think that's probably the most detailed starting point for people who are interested.
Yeah, the landing page for this webinar has some links on probably on the right side of your screen, I think. And one of those is for the Golden Lab. And if you go to the Golden Lab website, you can check out the SMART preprint is linked there. So, excellent. Thanks a lot, Sam. So, I mean, we've had a great talk here, and unfortunately, I think we're running low on time. So we'll answer one more question. It sort of follows up from the comments you just made about alignment. And I'm going to summarize a few different questions, but one is here from Ted Ustin. I think you know him at NIDA. He says, hello, Sam, beautiful talk and work. How closely do regions that are activated in various paradigms match atlas-defined cytoarchitectural regions? I think this question is similar to several others. Yeah.
That is a fantastic question. So in our most complete data set, which we feel we have all of the control groups and we have a high enough number of brains per group that we feel really comfortable with the data. We haven't done an unsupervised analysis yet, but it's on the list of things we absolutely want to do. Right now, I can tell you anecdotally, we see very, very clear areas of FOS that just map beautifully onto previously identified subregion architecture. We certainly also see brain regions where there seems to be a much more of a continuum of FOS activity that spreads around the classically defined regions. We have to dig into that, and we need to better understand that. Right now, I'm not really. I can say we see it. I don't know what it means. We certainly haven't taken advantage yet. I didn't talk about it, but obviously there are some really fantastic tools that allow us to manipulate behaviorally activated neural ensembles. In that case, for example, manipulating the neurons that are active during the behavior, which certainly means not just ones that are constrained to a specific subregion that we're targeting. And I think as we roll out those experiments, we'll be better able to say, what's the functional role of these populations that don't fall within these super well-defined, previously identified regions of interest, as identified by the ABA. We see them. We don't know what they mean yet. We don't know if it's one contiguous different region or if it's just maybe two adjacent regions that are both, we think they are when they're just both playing a role. These are all things we need to look into. Now that we have these data sets where we're not just looking at very specific subnuclei, individually. It's a great question. And I wish I had a more concrete answer other than to say, yeah, we really want to know what's going on with that. Can I answer
Excellent.
some questions? I see a couple that I like.
Oh, I'm fine with that. I still have time. I think if the Lab Roots team will let us continue a little bit longer, then you can spot the ones you're interested in answering among that list.
All right, there's a lot of technical questions here, which I'm not going to answer, but I think should have a discussion. So I'm just going to say, feel free to email me if we didn't cover your question, and I'll do my best to respond.
We'll give Sam this whole list, so everyone's list here. We'll work together, and we'll get everybody a response. So,
yeah. So I guess one that I'd really like to talk about that I don't have an answer to is sex differences in aggression. That's a really complicated answer because, for example, we've tried in CD1s and C56, so both outbred and inbred mice, female mice, to get them to lever press for aggressive interactions. And we've been unable to elicit aggressive behavior under those conditions. We can certainly train female mice to lever press for affiliative self-administration with a peer, where they can then, as mice, not as rats, but as mice interact, they will go on and show those types of social interactions. But we've not been able to do it with aggression. That said, obviously female mice show aggression under certain ethologically relevant conditions. Under the maternal conditions. Maternal aggression is a fantastic example. The Mitchell lab recently, Emily Newman, showed a procedure for eliciting robust aggression in female CD1s, as an example. It still requires some gestational components, but it's certainly a procedure that can be used to answer those questions. So that's something we're struggling with. And I would love to have a dialogue with the social behavior community on what is the most ethologically appropriate way to incorporate aggression research into sex as a biological variable. Because it's very hard to do since the mice don't natively do it on their own. And if you're artificially inducing it, is that, it certainly will identify mechanisms, but are those mechanisms one we really want to go after if they're artificial, things of that nature. And so I think that's going to be a real hot topic that the whole field needs to really try to address moving forward. One thing that we have been doing in collaboration with Emily is looking at the architecture of aggression between male and female CD1s. Are they similar? Are they different? I mean, is aggression the same behaviorally, or can we really reveal differences using both supervised and unsupervised approaches? And I think that's a good start. I think once we can get a hand on the behavioral differences in these models, then we can start getting a better idea of how to look at the neural similarities or differences that we can find. But it's a really complicated discussion because, again, in most of our models, that's like a royal us, it's very challenging to elicit aggressive behavior in females. And when it is elicited, it's elicited under very specific conditions to females. And so that's a really important discussion that we need to have and continue to have. It certainly has started, and then figure out what the neural, the underlying neural mechanisms are for these similarities and differences. Can I pick another one?
Sure. Thanks, Sam.
All right.
We appreciate it.
There's just a lot to pick from here, so I want to see if we can.
Yeah, we still have a lot of people hanging on the call, so really appreciate everyone's interest.
Yeah, so this is another really good general question, which again, I'm not sure I have an answer for, but what are the appropriate controls for whole brain data sets? When you're just looking at the nucleus accumbens, or you're just looking at the central amygdala, you, whether it's correct or not, often aren't viewing motor activity as something that needs to be controlled for as closely. We probably should be, right? But it seems like, oh, that's not a motor center, so we don't really need to maybe build in controls that take that into account. Whereas when we're doing something with whole brain, and we're getting everything in a completely unbiased manner across the motor cortex, the visual cortex, the auditory cortex, feeding into all of our subcortical regions, those questions become really important. What is our baseline? I don't have an answer for that. I see that question has come up a lot. I think what we need to do are design experiments with the most appropriate groups for the behaviors that we're really interested in. So for example, for a lever pressing behavior, where it's really hard, for example, to disambiguate the role of context to an operant chamber from the lever press itself. Right now, I think from these whole brain approaches, we're still going to get relatively messy data sets. But what we're going to be able to do with these data sets are start to go in and look at these non-canonical regions that seem to be activated that we haven't had a reason to look at before. And as we inactivate those or activate those using various approaches, we'll really be able to tie in a much more complete systems level understanding of what's occurring. It's a really hard question. It's a really good question. What are the appropriate controls? And I think it's very dependent upon the behavior and the designs that have been selected for the procedures that are being used with these whole brain data sets. But yeah, I mean, you get everything. You can't hide anything. If the whole cortex is lit up, it's very obvious. And then you need to understand, is that just because of the cue? Is that because of this? And using FOSS, that's not available yet. We don't have that temporal resolution for locking in truly discrete components of the behavior. And I'm only going to say we're very aware of this caveat, and there are active projects going on right now where we hope to be able to much more cleanly delineate the window of behavioral activity much, much better than what's available with current tools. But that's a story for probably another year, which hopefully we'll be able to share, but we'll see how that goes.
Excellent. Well, thank you again, Dr. Golden, for your time today and for all this important research. I think we're seeing, how your work with big behavior and big imaging, machine learning, this is all coming together and really taking advantage of the latest innovations and developments and technologies. So it's very exciting. I'd like to thank LabRoots for hosting this webinar, and on behalf of Miltenyi Biotech, I'd like to thank the audience for joining us today and for all these interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the registration. So we'll be forwarding all of those to Sam and as well. LabRoots will alert you via email when this webinar is available for replay. It may take about 30 minutes or so to have that process. It should be probably available later today. You'll be able to view it on demand if you had any technical difficulties this time. So until next time, I think that's all. And Sam and I will sign out, and hope you all have a nice day and stay safe out there.
Thank you.